large scale production of enzymes. Enzymes are the proteins from living cells that catalyze specific chemical reactions, that is, specific biochemical reactions. They may be intracellular or they may be extracellular, that is, secreted outside to prepare the nutrients. In general, extracellular enzymes are responsible for the degradation of complex macromolecules, whereas intracellulars are responsible for the regular metabolic processes, that is, simple carbohydrates, simple proteins, amino acids, that means structurally simple compounds. Enzymes are highly sensitive to change in pH, temperature, and other conditions like salt concentration, metal ion concentration, etc. There are six main groups: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. This part will be covered in the biochemistry course. Enzymes have many applications in different types of industries. That means enzymes are industrially very important. So the chemical reactions which are not possible or which are difficult or which are costly to be carried out using the synthetic processes. So these processes can be easily modified or they are catalyzed by means of enzymes. So commercially important enzymes or industrially important enzymes are amylases, proteases, pectinase, cellulase, collagenase, chitinase, glucose isomerase, glucose oxidase, lipase, invertase, lactase, penicillin acylase, pullulinase. You need to find out the substrates for all these different enzymes. For the production of enzymes, the selection criteria for the microorganisms for production or fermentation is the culture should be stable, possibly the enzyme should be extracellular so that its recovery and extraction from fermentation broth or downstream processing becomes easier. Microbes should give high yield of enzyme. The culture should grow on cheap substrates that will decrease the cost of fermentation. Formation of interfering substances should be low. That will indicate that will ease the way of extraction. There are several sources of enzymes like plants, animals, and microorganisms. Extraction from plants and animal cells is tedious because it is contaminated with several other proteins, and therefore the most reliable one is microorganisms. So this table shows the applications of commercially important enzymes. This is given in the book of Principles of Fermentation Technology by Stanbury Whitaker. So the first column indicates the type of industry in which the enzymes are utilized. Second column is application. Third column is the name of the enzyme and its source. So we'll see few examples. So amylases are produced from bacteria as well as fungi, and they have applications in baking and milling industry. So basically their role is to cause reduction of duff viscosity while making of bread. Acceleration of fermentation, increase in loaf volume, so the sponginess increases. Improvement of crumb softness and maintenance of freshness. So amylases are involved in this application while making of bread. So it is responsible for the modification of duff texture. It is also useful in during the mashing process. Amylases are useful in mashing during the beer making process. So mashing is the softening of mixture in which the barley grains are milled, and this barley is having high concentration of starch which has to be hydrolyzed. So barley malt is contributing amylases which is responsible for mashing. Then amylases are also useful in the laundry industry where the detergents uh, are used. So detergents are having a concentration of amylases, and amylases are used along with proteases in detergents as well as lipases. So these are very useful for the removal of food spots. Then amylases are useful for as a digestive aid. That means they are having pharmaceutical use. Then in textile industry for the sizing of fabrics, amylases are used. Other applications are in chocolate, cereal, chocolate, and cocoa coffee industries. Amylases are basically useful for the treatment of baby foods. So, in order to make it more digestible, the starchy substances are degraded by using amylases. Making of syrups, coffee bean ferment, making of syrups and breakfast foods. These are the various applications of amylases. We'll see the production of amylases in detail. So these are the uses in textile industry: preparation of starch paste, degradation of starch in the making of chocolate syrup and corn syrup, in baking industry, in dry cleaning industry, along with proteases, and the pre-treatment of the raw materials like in case of mashing during the making of beer. The producer organisms are bacillus species, so several vast number of microorganisms are known for the production of amylases. Uh, Among fungi, commercial strains are Aspergillus oryzae and Aspergillus niger. The methods of production are submerged method of production and solid state fermentation by using thin layer technique and deep bed process. The production of fungal amylase by solid state fermentation. The enzyme, this fermentation has this following advantage: that enzyme yield per unit volume is high, power requirement is low, there is minimum control necessary, small equipments are required for recovery, and scaling up is easy. These are the advantages of production of fungal amylase by solid state fermentation. For this, the organism used is Aspergillus oryzae, and there are two techniques: thin layer technique and deep bed process. In thin layer technique, a substrate is layered with thickness of 2 to 4 centimeter in wooden or metallic trays. Then sterile wheat bran or rice bran along with soybean meal, sweet potato flakes may be used in addition to the trace elements such as iron, zinc, copper, and manganese in the form of salts. So all these metal ions are required for the germination of spores. So this is the fermentation which is carried out in the absence of free water, that is solid state fermentation. Aspergillus oryzae, it is cultivated for the development of spore inoculum. For the medium is acidified. So in most of the fermentations which are carried out by fungi, the pH of the medium is acidic. This is followed by the inoculation with the spores of Aspergillus oryzae. Then the trays are incubated for 4 days at a temperature of 20 to 25 degrees Celsius. There is generation of heat during fermentation, which is controlled by passage of cool air, that will serve for the two purpose, temperature control as well as providing aerobic conditions. This is the flow sheet. This is the wheat bran solution, which is sterilized in a steam cooker. Then it is acidified, and then it is spreaded into the trays in thin layers and then placed into cultivation tunnel, which are then inoculated with the spores of Aspergillus oryzae. After the incubation period of 3 to 5 days, then these trays are ready for extraction and purification, or ready for downstream processing of amylase. So first step in that is these trays can go directly to drying tunnel, where the contents are exposed to temperature less than 50 degrees Celsius, so that the water will be evaporated. This is a method of concentration. After drying, after dewatering process, the contents will go to a grinder, so that a uniform homogeneous particle size preparation will be produced, and then this ground material will go to an extractor, where this extractor has a strainer, and there is addition of water. So this content will be added into the extractor. This is a strainer, that means it will act as a filtration device. Then there is addition of water, and this is how the contents are extracted by using water. So finally we get the filtrate, and the solids will remain in this device. So this preparation, that is the fermentation broth, which is free from solids, is then ready for further processing. Next step is precipitation. So considering that amylases are proteinic in nature, the best way for separation of proteins like amylases is precipitation by using salts or solvents. So salts like ammonium sulfate, it can be added to this filtrate, or there can be addition of acetone or ethanol. Now here the concentration of salt and solvent is very important. You are familiar with the concept of salting in and salting out. So it is already studied that at what concentration of salt and solvent this desirable amylase is precipitated. So that concentration of salt and solvent should be added to this filtrate, so that the amylase having that particular property will be precipitated, and rest of the proteins will remain solubilized. After precipitation, the precipitate is then separated from the soluble impurities. Now here most of the soluble proteins will be separated, and this will be this will lead to the concentration. Then this precipitate is separated, amylase enriched precipitate, and this precipitate is then further uh, go for the next step of concentration, that is ultrafiltration. Ultrafiltration is a selective technique for purification of biomolecules that separates the biomolecules on the basis of its molecular size, that is ultrafiltration. So if you are looking for specific size of amylase, molecular size of amylase, then accordingly that cutoff size ultrafilter membrane should be used, and that will lead to the separation of desirable sized amylases from rest of the undesirable proteins. Further, the purification is done by using chromatography techniques using different columns. It also includes the ion exchange chromatography depending on the nature of amylase which is to be separated. After chromatography, then again the next step is precipitation. So the step of precipitation is again used to minimize the impurities. Further, drying, depending on the thermal characteristic of amylase, the temperature is used. So drying in evaporator, and finally it will go to cold storage until the packaging is done. Now the production of amylase by submerged fermentation. Production of fungal amylase by submerged fermentation. Here a different strain is used, that is Aspergillus niger. First step is the inoculum preparation. The stock culture is inoculated in sterile nutrient solution with continuous aeration and agitation for 3 to 4 days till the sporulation occurs. Production is done in the production fermenter which is well equipped. The production medium consists of molasses or barley or wheat or rice, corn, bread with presence of excess water, because this is submerged fermentation. Different types of salts and other trace elements should be present in production medium. The incubation temperature is 20 to 25 for 3 to 4 days with continuous aeration and agitation. The recovery, it starts with the separation of mycelia by filtration or centrifugation. Then this broth is chilled at 5 degrees Celsius to avoid the inactivation of amylases which may happen. The separation of colloidal material is facilitated by addition of coagulating agents such as diatomaceous earth. So solids are separated by using this filter aid. Then the further steps are the same: precipitation, that is treatment with alcohol for the precipitation of proteins, that is enzymes, and undesirable proteins will remain soluble, which can be separated. Next step is filtration, concentration by ultrafiltration, purification by column chromatography, final precipitation by ammonium sulfate or acetone, and then drying in a spray dryer or freeze dryer. If the amylases are thermostable, then spray dryer can be used. If it is not, then freeze drying, that is lyophilization, can be done, and then packaging. This is the flow sheet for submerged fermentation Aspergillus niger. The spores are inoculated. So amylases are extracellular and primary metabolites. Then filtration, centrifugation, initial precipitation, separation of precipitate by centrifugation, ultrafiltration, chromatography, collection of fractions, final precipitation, and freeze dry.